So hi everyone, today we'll be discussing the most important topic in which most of the exams these questions were asked regarding this topic, that is the glycogen storage diseases. There are generally 6 types of glycogen storage diseases. We'll go in with each of them, but first of all we'll start with the most important glycogen storage disease, that is type 1 glycogen storage disease. Type 1 glycogen storage disease, which we also call it as Guy's disease. So this disease is very important because there are many pathologies that are related with this disease. We will discuss each of the pathology now. So first of all, this disease occurs by the deficiency of the enzyme glucose 6 phosphatase. So when you take your liver cell, right? Suppose this is your liver. As I told you, in liver there is excess amount of glycogen, right? So I'll simply write it as GLY, excess amount of glycogen. And in the extracellular fluid you find many proteins including glucose, GLU, right? So the question is that what is the chief source of glucose in the liver? There are two ways. One is that the glucose by itself from the blood enters into the liver. This is one chief source. And the next is that glycogen converts into glucose 6 phosphate or glucose, simply glucose, right? So what exactly happens is that glucose can easily cross any membrane and enter into the cell, and can easily cross any membrane and come out of the cell. So glucose can easily enter into the cell, right? When there is a deficiency of glucose, obviously the glucose which has entered may easily come out of the cell. But this not happens here. What exactly happens here is the glucose which is entered into the cell, it is trapped by this liver. How it is trapped by the liver? Immediately when the glucose from the extracellular fluid enters into the liver, glucose is added with a phosphate, so that glucose becomes glucose 6 phosphate, and that glucose 6 phosphate cannot be, or it cannot exit out of the cell, right? So glucose is added with a phosphate, thus it forms glucose 6 phosphate, G6P. Sometimes glycogen can directly be converted into glucose 6 phosphate, right? So finally glucose 6 phosphate is formed. So as you know, liver, any cell contains endoplasmic reticulum. Or suppose we take the endoplasmic reticulum, we enlarge the endoplasmic reticulum here. As you know, endoplasmic reticulum has two membranes. This is the outer membrane and this is the inner membrane, right? This is the outer membrane and this is the inner membrane of the endoplasmic reticulum. So this becomes your cytosol and this becomes your endoplasmic reticulum. What exactly happens is, on the surface of endoplasmic reticulum you find protein molecules, mainly transporter proteins. These transporter proteins are named by the substance that enters through it. For suppose this glucose 6 phosphate is entering through these transporter proteins into the endoplasmic reticulum from the cytosol. So this transporter protein is named as glucose 6 phosphate transporter protein, right? So this transporter protein is named as glucose 6 phosphate transporter protein number 1. Glucose 6 phosphate transporter protein number 1. So through these transporter proteins, this glucose 6 phosphate enters into the endoplasmic reticulum. So this is glucose 6 phosphate which has entered into the endoplasmic reticulum through this transporter protein. So as I told you, this is cytosol and this is endoplasmic reticulum. We are talking about the single cell in a liver, right? Now, on the inner membrane of the endoplasmic reticulum, this is outer, this is the inner membrane. On the inner membrane of the endoplasmic reticulum, there is an enzyme which we name it as glucose 6 phosphatase, right? This is an enzyme called as glucose 6 phosphatase, which is bounded or which is situated on the inner membrane of endoplasmic reticulum. The function of this enzyme is it converts the glucose 6 phosphate which entered into the endoplasmic reticulum into two components. One is glucose and another one is inorganic phosphate. So whenever there is hypoglycemic condition, whenever there is low amount of glucose in our blood, immediately the glucose 6 phosphate enters into the endoplasmic reticulum, and by the action of glucose 6 phosphatase, it converts into glucose and inorganic phosphate, and this liberated glucose exits out to again normalize the level of glucose in the blood, right? So what exactly happens in Gyrech's disease? What exactly happens in Gyrech's disease? As I told you, there is a deficiency of the enzyme glucose 6 phosphatase. So there are two types of Gyrech's disease. One is type 1A, type 1B. Type 1A and type 1B Gyrech's disease. What is type 1A Gyrech's disease? If there is a problem with this glucose 6 phosphate transferase protein number 1, then obviously glucose 6 phosphate will not enter into the endoplasmic reticulum. When it does not enter into endoplasmic reticulum, it cannot be converted into glucose and inorganic phosphate, and there will be no longer supply of glucose from the liver into the bloodstream. So this is type 1A of Gyrech's disease. Next, type 1B is this one, the enzyme. Type 1B. If there is a deficiency of this enzyme, then glucose 6 phosphate will no longer be converted into glucose and inorganic phosphate. So there is accumulation of glucose 6 phosphate within the cell. This accumulation causes many diseases or many pathologies, and that is the reason we call it as Gyrech's disease, right? So now we will see what are the pathologies that are caused by the accumulation of glucose 6 phosphate. So I'll just rub it and draw it in another pattern. So just now, as you saw, glycogen, GLY, right, in abbreviated form. It's okay, glycogen. Glycogen is converted into glucose 6 phosphate, right? And next, glucose 6 phosphate, with the help of the enzyme glucose 6 phosphatase, right? Glucose 6 by the enzyme glucose 6 phosphatase, glucose 6 phosphate is converted into glucose, right? And inorganic matter, that is inorganic phosphate, that is another concept. So it is converted into glucose. Now when there is a deficiency of this glucose 6 phosphate, what happens? Glucose is not formed, but glycogen converts into glucose 6 phosphate as usually. But from glucose 6 phosphate there is no glucose. So what you need to understand is that the concentration of glucose 6 phosphate is keeping on increasing, right? And one more thing, not only glucose is formed, glucose 6 phosphate is also a substrate for the formation of ribose 5 phosphate by a pathway called as pentose phosphate pathway, PPP, pentose phosphate pathway. By this pathway, glucose 6 phosphate is converted into ribose 5 phosphate, ribose 5 phosphate, right? Another pathway, glucose 6 phosphate is converted into pyruvate, right, by glycolysis. By glycolysis, glucose 6 phosphate is converted into pyruvate, and further pyruvate is converted into acetyl-CoA, and this acetyl-CoA enters into the Krebs cycle, and finally it forms fatty acid, glycerol, as well as cholesterol, and that is another pattern I will be discussing in a while. So glycogen is converted into glucose 6 phosphate. Glucose 6 phosphate is acting as a substrate for the formation of glucose, Glucose 6-phosphate is acting as a substrate for the formation of pyruvate through glycolysis, and glucose 6-phosphate is acting as substrate for the formation of ribose 5-phosphate through pentose phosphate pathway. Now, if glucose 6-phosphatase, if there is a deficiency of this glucose 6-phosphatase, what happens? Glucose is not formed, right? So more and more glucose 6-phosphate which is formed, basically more and more glucose 6-phosphate which is formed is equally distributed into 3 of these cycles for the formation of their products. But if due to this enzyme deficiency, more and more glucose 6-phosphate is accumulated, right? More and more glucose phosphate is accumulated and it is shunted for the production of pyruvate and ribose 5-phosphate in excessive amount. So when more and more G6P is accumulated, it forms more and more ribose 5-phosphate, excess amount, and even it also forms more and more pyruvate. When more and more ribose 5-phosphate is formed, automatically ribose 5-phosphate undergoes catabolism and forms uric acid. When more G6P is there, more uric acid is formed. When less G6P, less uric acid. But in this pathology, there is more G6P, so more uric acid is formed. This is called as hyperuremia. So this is one pathology of the Gyrix disease. Now coming to the next. As I told, when glucose 6-phosphate is absent, then there is no formation of glucose. So when there is no formation of glucose, then the glucose blood level is decreased, which we also call as hypoglycemia. So this is the second pathology related with Gyrix disease. Now coming to the third. More and more G6P converts into more and more pyruvate, right? More and more pyruvate converts into more and more acetyl-CoA. When more and more acetyl-CoA is formed, this more acetyl-CoA, excess amount of acetyl-CoA enters into Krebs cycle and finally results in the formation of cholesterol and fatty acids, right? These fatty acids suddenly or immediately it combines with glycerol. So cholesterol, fatty acids and glycerol combinedly, there is a pathology that is called as hyperlipidemia. It results in the formation of hyperlipidemia or hyperlipidemia, right? So so far we have seen 3 pathologies. Now still there are more. As I told, pyruvate, as it is keeping on increasing, it forms acetyl-CoA, right? Not only acetyl-CoA, pyruvate also takes part in the formation of an important amino acid called as alanine. Right? If more pyruvate is formed, more alanine is formed. So this is called as hyperalaninema. So spelling hyperalanemia or something like that, hyperalanemia something, right? Next, pyruvate not only forms alanine, but pyruvate also forms lactic acid or lactate. So more lactate, more G6P, more pyruvate. When more pyruvate, more lactate. When there is more lactate, it is called as lactic acidosis. Lactic acidosis. So 1, 2, 3, 4, 5. Mainly 5 pathologies. There are still many pathologies that are related with Gyrix diseases, but principally we find 5 pathologies. So once again a quick review. As you know, glycogen is a chief source which is present in the liver. Glycogen is converted into glucose 6-phosphate. Glucose 6-phosphate which is formed is equally distributed to form pyruvate, is equally distributed to form glucose, and is equally distributed to form ribose 5-phosphate. So if glucose 6-phosphatase, which is used to convert glucose 6-phosphate to glucose, if this enzyme deficiency occurs, then there will be more pressure for the production of pyruvate as well as ribose 5-phosphate. So obviously glucose 6-phosphate will be keeping on accumulating in the liver, and pyruvate will be formed in excess amount. In the same way, ribose 5-phosphate will be formed in excess amount by pentose phosphate pathway. When excess ribose 5-phosphate, it undergoes catabolism to form uric acid. More uric acid called as hyperuremia. Coming to pyruvate, more G6P, more glycolysis, more pyruvate. When more pyruvate, more alanine called as hyperalanemia. More pyruvate produces more lactate called as lactic acidosis, right? And next, pyruvate further converts into acetyl-CoA, and acetyl-CoA enters into the Krebs cycle. The final products of Krebs cycle are cholesterol, fatty acids, and these fatty acids combine with the glycerol. So combinedly a pathology called as hyperlipidemia, and this is hypoglycemia. So these are the pathologies that are related with the Geirex disease. So the examiner can ask any one of them. So they may also ask lactic acidosis is a characteristic feature of which disease. He may ask type 1, type 2, type 3, type 4, or he may also give you Geirex disease or so and so diseases which are related with this glycogen storage. So this is the important glycogen storage disease among the 6 types. So now remaining glycogen storage diseases, you no need to remind the entire concept. You just need to remind what are the enzyme deficiencies that are present in those diseases. That's it. So we'll have a quick review overall on these GSD, glycogen storage diseases. Type 1 disease, as I discussed. Type 2 disease. Type 3. Type 4. Type 5, and the last type 6. So there are 6 types of glycogen storage disease. What is type 1? As I told you, type 1 is Geirex disease. Type 2 is Pompey's disease. It is named after a gentleman who proposed this disease or who invented this disease, who discovered Pompey's disease. Type 3 is Forbes, it is something. Yeah, Forbes disease. It is Forbes disease. And type 4 is Anderson disease. Anderson's disease. And type 5 is McAlder's disease. And the final disease, that is type 6, is Hers disease, HERS, right? Now we will discuss what are the enzyme deficiencies of each disease. Geirex disease is due to the deficiency of the enzyme glucose 6-phosphatase, right? Is Pompey's disease by acid alpha glucosidase. Acid alpha glucosidase causes Pompey's disease. And Forbes disease, it is amylo 1,6 glucosidase. Amylo 1,6 glucosidase. And Anderson disease is amylo, the same as Forbes disease, amylo 1,4,1,6 trans glucosidase, right? And next, Alderson disease is myophosphatase. And at last, Hus disease is liver phosphatase. So these are the enzyme deficiencies which you need to remember. And mainly, as I told you, Geiger's disease is most important, which you need to remember the entire concept. But these diseases, you just need to remember their respective types and respective enzyme deficiencies. And one important thing which I need to discuss regarding this type 5 disease or McAlder's disease is, muscle cell contains glycogen, as I told you. Glycogen must be converted into glucose by the action of this enzyme myophosphatase. When this enzyme is absent, glycogen will no longer be converted into glucose. So accumulation of glycogen causes the breakdown of the muscle proteins. When muscle proteins get broken down, they release small particles called as myoglobin particles. These myoglobin particles can be found when they enter into the blood. It is very dangerous because they may damage your kidney. So it may also cause renal failure. That is the reason why patients with McAlder's disease, in their urine you find red color molecules called as myoglobin molecules. And the urine of these McAlder's patients looks red color, which we also call it as red urine. And the pathology related with this McAlder's disease is myoglobinuria, right? So this is the one thing which you need to remember, and these are the glycogen storage diseases. So next storage diseases will be discussing regarding the protein storage diseases and lipid storage diseases. Thank you.